direct reply after the live webinar. At this time, all participants are in listen-only mode. This event will be recorded and made available for future download from www.bendresearch.com. The presentation slides will advance automatically. If you need technical assistance, please contact GoToWebinar at the numbers displayed. In the U.S., 800-263-6317 or 1-805-617-7000. Now we'd like to begin the formal presentation. Bend Research is a leading independent scientific development and manufacturing company. For more than 35 years, our multidisciplinary teams have pioneered and advanced technologies to solve our clients' most challenging technical problems. The success of Bend Research is based on our strong understanding of science and engineering fundamentals and extensive expertise in materials science, process engineering, and applied mathematics and modeling. Bend Research has numerous capabilities to support the advancement of promising drug candidates from conception through commercialization. These include formulation and dosage form support, process development and optimization, and cGMP manufacturing. We are a leader in novel drug delivery technologies such as spray drying, hot melt extrusion, and controlled release, and we have expertise in inhalation and biotherapeutics development. Bend employs nearly 300 people and has 6 state-of-the-art facilities in Bend, Oregon. I'd now like to welcome our speakers for today's seminar, Lisa Graham, Ph.D., PE, and Brandon Downey. Dr. Lisa Graham, Senior Vice President at Bend Research, is responsible for business development, particularly for biotherapeutics and new technology areas, the management of major client relationships, strategic planning, and technology and engineering development. Dr. Graham's areas of focus include growth of the biotherapeutics business programs for large molecules and leveraging technical expertise in the application of fundamental engineering knowledge to the development of models and other tools. The goals of these tools and models are to promote in-depth understanding of pharmaceutical unit operations, to promote efficient scale-up, scale-down, and technology transfer for small and large molecules. She received a Bachelor of Science in Chemical Engineering and a Ph.D. in Chemical Engineering, both from Oregon State University. She is a licensed professional engineer in Oregon, the author of 10 scientific papers, and a member of the Oregon Institute of Technology President's Advisory Council, the Oregon State University Chemical, Biological, and Environmental Engineering Industrial Advisory Board, the American Institute for Chemical Engineers, the Oregon University Systems Engineering and Technology Industry Council, and the Professional Engineers of Oregon. Dr. Graham has been with Bend Research since 2002. Brandon Downey is a research chemical engineer and technical group leader at Bend Research, where he works on new technology development programs. His primary areas of expertise and responsibility include the development, scale-up, and integration of new manufacturing technologies into biotherapeutic production processes. He has worked extensively with dielectric spectroscopy probes applied to monitoring and control of mammalian cell culture processes. He earned his Bachelor of Science in Chemical Engineering from the University of Colorado in Boulder. And now, without further ado, I'd like to hand the microphone over to Lisa Graham. Lisa, you may begin when ready.
Thank you, Donna, very much for the introductions, and thank you to everyone who's taken time out of their day to call in for our technical review of dielectric spectroscopy. We'd like to start by putting up a brief outline. These are some of the key areas that we're going to focus on this morning. Brandon will go through the fundamentals of dielectric spectroscopy and a couple of case studies, and I'm going to take up the baton at the end to talk about how dielectric spectroscopy fits into the broader scope of all the other tools that we use. So we'd like to start with a single slide that attempts to put dielectric spectroscopy in context for why it is useful in bioprocess development. In preparing this presentation, we did a bit of work to understand why scientists might be trying to leverage a tool that provides real-time observability of what's happening in the bioreactor. As many of you likely know, DS probes are currently used quite extensively across the lab, pilot, and commercial scales. And for example, having a tool in the earlier cell line development stage or early lab stage can open the door for several different opportunities for new process development methodologies. In later stage development at the pilot and commercial scale, it can lend itself to innovative process monitoring and control strategies. As we've been using the technology for a number of applications over the last several years, we've realized how important it is for our scientists to really understand the fundamentals so that we can leverage the technology in a number of applications. And with that in mind, I'm going to turn the presentation over to Brandon so we can get into the technical content of this program.
All right, thank you very much, Lisa, for the introduction. So we're going to start off by just spending about 15 minutes and going through the fundamentals of dielectric spectroscopy to give a little more background on the technology. And then we're going to go through a couple case studies where we've actually found the technology itself to be useful in practice. Those that are familiar with the technology probably know it in the context of measuring biomass as a tool for measuring biomass in situ in the bioreactor. And so the reason we can do this is that we're taking advantage of the fact that cells act as capacitors under the influence of an electric field. So you can therefore imagine that the more number of cells that we have in the bioreactor, the higher total capacitance that we ultimately measure. So as we mentioned before, briefly, the dielectric probes collect data by sending out an electric field into the bioreactor and measuring the electric response, which is induced by these charges that form on the cell membrane in the presence of an electric field. Today we're going to review not only this biomass application where we care about the total single frequency capacitance of the system, but we're also going to review about techniques where we care not only about the number of capacitors, but also the characteristics of how these capacitors charge differentially from each other. So in order to understand the technology a little more, we're going to spend about 15 minutes going through the fundamental physics of the technique, and then we'll get back to more of the application side of things. So we mentioned at the beginning of the talk the cells act like capacitors under an electric field. And so really, what does this mean in terms of basic physics? So a capacitor is just a device that stores charge. A man-made capacitor, such as the example shown schematically here, can be described by the figure on the bottom left. This capacitor consists of two conductive plates, which are shown in red, and these are connected in a circuit, which are denoted by the black lines. So sandwiched in between these plates is an insulating material called a dielectric, and that's shown in light yellow. And this dielectric actually helps the capacitor to store charge. So we're going to get to dielectrics in a minute. The point of this slide is that the cell can actually be thought of as an analog of a man-made capacitor. So this is shown in the figure to the right. So any isolating membrane, such as a cell containing ions, will act somewhat as a dielectric via this polarization at the interface. And this is known as Maxwell-Wagner polarization. So one other point to make here is that the total amount of charge a capacitor can store, which is defined as the capacitance, which is shown as C in the equation here, depends on both the geometric properties of that capacitor, which are the A and D terms, as well as the properties of the dielectric that's in between the plates, which is known as the permittivity, and that's denoted by epsilon. So capacitance and permittivity are therefore directly proportional, and we're actually going to consider those to be equivalent measures throughout the rest of the talk. So we saw on the last slide that one of the major factors in determining how well a capacitor stores charge are the properties of that dielectric material. So if we care about how well a capacitor, or a cell in our case, stores charge, then we by default care about dielectrics. So a dielectric is essentially an insulating material that contains a bunch of dipoles. And these dipoles are, under normal conditions, randomly oriented, so under zero energy conditions. So this is like having a block attached to a wall by a spring, but under normal conditions, the spring is relaxed. So the dipoles can be thought of in this way. Now, if I force all the dipoles to face a certain direction, that takes energy. And so that's like pulling back on the block attached to the spring. So obviously now if I were to let go of that block after I pulled it back away from the wall, the block would then return back to its starting position. So similarly, if I release the field that's orienting the dipoles in this way, the dipoles will actually go back to their randomly oriented ways, and I'll get back that energy that I put in to orient them in the first place. So another aspect of dielectrics that we're not going to deal with here, but I'm just going to mention, is that you also lose some energy in this process as well. And you can actually quantify this. It's quantified by a term known as the dielectric loss. It's sort of like adding friction to the spring analogy that we just discussed. So how do we go about measuring these properties of dielectrics? So it turns out if we subject a capacitor to an alternating electric field, it acts as a special circuit, which is known as an RC circuit, which is shown here. So in the RC circuit, we have both an ideal energy storage component, or a capacitor, which is shown in red, and an ideal loss component, or a resistor, which is shown in green. So since we're surmising here that the cells act like capacitors, they will also behave as an RC circuit in this alternating field. So why use an alternating field? One of the nice things about alternating fields is that we actually know exactly how an RC circuit will respond to a field of this type. And then we can easily measure the result of the response. So if we send out an alternating field, which is labeled as the driven field here, the sine wave in blue, the cells will produce their own field due to polarizing in response to the driven field, and that can be detected, and that's known as the response field, and that's the red sine wave. So we can characterize the differences between the driven and the response fields by measuring both the magnitude change, which is solely due to the loss component of the RC circuit, and by measuring the phase lag between the two sine waves, which is due to the storage component. So now we have this convenient way of measuring dielectric responses in cells. How do these responses change if I scan across the frequency spectrum? So if we plot the frequency-dependent behavior across the frequency range, we get a characteristic shape which depends on the dielectric properties of the capacitor population that we're measuring. And this behavior is known as the beta dispersion. So to understand why we get this behavior, we're going to use a sort of a cheesy but hopefully instructive analogy of dielectric spectroscopy as a molecular limbo contest. So in our molecular limbo contest, the dielectric storage component, or the capacitance or permittivity here, all equivalent, can be thought of as the active number of contestants still in the contest. And the dielectric loss component can be thought of as the number of contestants which are subsequently eliminated through each round. So to step through it, in order of increasing frequency, at very low frequencies, we observe this high frequency plateau in the capacitance, and we observe almost no loss. And so physically, that's due to the fact that we're moving this field back and forth so slowly that really everything that's going to polarize has a chance to polarize. So we get a very high storage component and almost no loss. So in terms of back to our limbo analogy, that's like saying we've raised the bar so high that we're really not eliminating anyone from the contest. So we're not really learning anything about anybody's limbo abilities with this. But as we start increasing the frequencies to higher and higher frequencies, that's essentially like incrementally lowering the bar in the limbo contest. So you can see that we get this characteristic decrease in the storage component, so we're eliminating more and more contestants as it gets harder to get under the bar. And we kind of go through a maxima in the loss where the majority of people are eliminated kind of halfway through the contest, but there's some elite people that make it all the way to the end. So back to dielectrics, that's we're only polarizing the things that are very good at polarizing at the very high end of the frequency range. So now we're going to get to why we care about this in terms of cells. So if I have a single population of cells, and this is some population where I can define that population in terms of some dielectric property that's different among the cells, I can observe the case that we just discussed in the last slide. So that's like saying I have a single distribution of limboability on my limbo team. And so this behavior is described in the graph below here, and that's known as a single-term Cole-Cole model. But perhaps the more interesting case is what happens if I now have two cell populations with different electrical properties. So that's like saying I have two limbo teams of different limbo abilities. So one of those teams is actually going to get further along in the contest than the other team is. And so in this situation, we're actually going to see multiple relaxation properties, as shown here. So if the teams are very poorly matched, one team would be pretty much totally eliminated before the other better team even started to lose contestants. So this is described by a multiple term Cole-Cole model, which is shown on the figure on the lower right. So it's worth noting that in practice, the teams are not this poorly matched usually, in terms of actual cells, that is. So we observe in practice a much more subtle effect than this. This is just drawn starkly so we can see the trend. So we've seen how having multiple populations of dielectrics or cells or limboers can be detected by dielectric spectroscopy using frequency scanning. We're going to wrap up the fundamental section by just mentioning that this has all been observed in the literature using dielectric spectroscopy on cells. So there are specific references which have included here where the authors of these references have manipulated the dielectric properties of cells and observed a resulting change in the dielectric spectrum. And I can provide that reference list to anyone that's interested after the talk today. So now we're going to kind of move into our first case study to talk more about how we can use this information in practice. So in this case study, we're going to talk about how we can use frequency scanning shape information to predict a viability-based divergence that occurs between viable cell mass as measured by total capacitance and viable cell mass as measured by an offline method, such as trypan blue dye exclusion. So if you're familiar with capacitance probes at all, you may have noticed the phenomenon which is depicted here. So what's shown in the graph is an apparent viability-based divergence between total capacitance and the viable cell volume as measured via some offline cell counting method, such as trypan blue. So you can see that in the growth phase of the culture, which is up to about 150 hours in this data set, that we can use the total capacitance, or permittivity in this case, pretty reliably as an indicator of the actual offline viable cell volume, or at least as well as the offline method can determine the viable cell volume. As we enter the death phase of the culture, however, you can see that we begin to largely overpredict the viable cell volume using the total capacitance value. So the same phenomenon, the same data set viewed another way here shows how total capacitance, or permittivity label here again, is correlated to the viable cell volume measured using the offline method. So we get a nice linear correlation in the growth phase again, but diverge from that correlation during the death phase. So this is data that was actually generated here in Bend on an N0 cell line. But this same phenomenon has actually been observed in other published data sets in the literature as well, in other mammalian cell lines. So this is an example from a CHO line, and you can see that this is a pretty robust phenomenon. So we're going to show in this case study how you can actually go about using frequency scanning to correct for this divergence and end up with a result similar to that shown here. So if you've observed this phenomenon in-house, you may have noticed just by sort of playing around with the numbers that you can compensate for this divergence largely by just subtracting out some part of the observed signal. So essentially just forcing all those points to fall back onto the line. And you may have also noticed that the amount of signal that you need to subtract appears to be proportional to the measured offline viability of the cells at that point. So this relationship is formalized in the equation shown above here in the green box, where I can obtain a corrected signal by taking the raw capacitance or permittivity signal and subtracting some portion of that signal, which is determined by a correction factor that's shown as Fp of t there. So this correction factor we found to be inversely related to the percent viability of the culture as measured by trypan blue. So this fact is useful in understanding that the divergence may be related to the viability of the culture, but it kind of defeats the whole purpose of having a totally online measurement if we need to take offline samples to correct the online measurement. So the question we asked was whether or not there was a way to determine this correction factor, Fp of t, using the information contained in the frequency scanning information at each of those time points. So our working hypothesis to explain this divergence is that there might be some electrical properties of the cell population which are related to either cell health or cell death, and therefore those properties must be changing once the cell health begins to change. And those properties are being detected by dielectric spectroscopy. So if you look at the pictures of the actual cells during the growth phase and the death phase, this sort of makes intuitive sense. We see the divergence occur when we observe unhealthy cells. So again, can we use frequency scanning to quantify these differences in the electrical properties between the healthy and unhealthy cell populations? So we took frequency scanning data throughout a cell culture run, and representative dielectric spectra are shown here at 4 representative time points throughout the run in the graph on the right. So here we're looking only at the dielectric storage component versus frequency. So you'll notice that the curves in this case are all at different heights, and they may or may not be different shapes. So the fact that they're all at different heights is due to the fact that we have a different number of total cells at different points throughout the culture. So if we really want to get a good idea of the shapes of the curves relative to each other, we have to take the total number of cells out of the equation because, again, we're trying to look at some other electrical property of the cells besides cell number in this case. So to better visualize that, we removed the confounding effect of cell number by normalizing each curve from 0 to 1. So we did this here by just dividing each capacitance value observed at each frequency along the entire frequency scan by the capacitance measured at the lowest frequency. So when we do this, we observe sort of an interesting pattern, which is shown in the graph on the right there. So during the growth phase of the culture, the normalized curves, which are labeled as blue and green here, are actually so similar in shape that they essentially lie right on top of each other. But as we move into the death phase of the culture, however, we begin to diverge from that initial shape of the growth phase curve progressively over time. And so these death phase curves are in order yellow and then red later and later in the run. So initially it looks like we're supporting our hypothesis. A stable dielectric shape, or stable shape of the beta dispersion rather, is observed throughout the growth phase of the culture, which is when we observe healthy cells. But as we go and proceed into the death phase, our spectrum moves away from that original stable shape, and that's when we observe unhealthy cells. So if we want to actually use this shape information, we have to quantify it somehow. We have to quantify the shape change that's occurring over time. So to do that here, we use the simple area ratio. So in the ratio, we take the area under the whole of each curve divided by the area under just the upper portion of each curve, or the higher frequency portion of each curve. And that way we've reduced the entire shape of the curve to a single number. And the mathematical formalism of this is described below. So if we take that area ratio number now, which again is only a single number describing the shape of the dielectric spectrum changing over time, we get the curve which is shown on the right. So the first thing you'll probably notice is that this looks suspiciously like the inverse of a viability curve for this culture. And remember we said at the beginning of the talk that our correction factor appeared to be inversely proportional to viability. So far it's looking good. So we attempted to correlate the area ratio to the observed percent viability for this culture and actually ended up with a pretty decent correlation, and that's shown on the left there. The graph on the right now shows the viability that's actually predicted by that correlation, which is shown as the black line, and the actual data from the CDEX is shown by the red squares. So it looks like we can predict the percent viability of the culture using the shape information that's contained in the dielectric spectrum that we've obtained from frequency scanning. So now we've established we can do that, the question is, can we use this information to correct for this death phase divergence between the capacitance and trypan blue methods? So since we sort of gave away the punchline at the beginning, the answer is yes in this case. So shown here is the data from our cell culture run showing the correlation between, again, total permittivity or capacitance and the viable cell volume using measured in the offline method. So here the only difference is we split the data up separately into growth phases, which is the growth phase shown in green is the linear portion of that curve, and then the death phase, which diverges from that growth phase curve. So if we employ the area ratio to predict our correction factor from the beginning of the section, which is FP of t again, we can get all of the points to fall back onto that growth phase correction line. And now if we use this new correction method now to predict the viable cell volume, we can get the following results shown on the lower right. The original uncorrected values are shown in blue, and the corrected measures are shown in green. So a pretty interesting result. One thing that's worth noting about this result is that you'd have to perform an initial correlation building run first to determine the relationship between the area ratio and the correction factor, and then you could use that correlation on subsequent runs to perform this correction in real time. So here we've demonstrated a way to use the information obtained from frequency scanning to enhance our measurement of viable cell volume by capacitance in the death phase. So having a method to measure this viable cell volume accurately during all phases of the culture enables, obviously, you to obtain an accurate viable cell volume measurement throughout the run without having to sample. But perhaps more interestingly, it also allows you to implement a feed strategy based on biomass without having to worry about overfeeding later in the run or having errors in feeding. And obviously, we'd want to integrate this into an advanced control scheme. So now we're going to move on to the second case study, which involves using frequency scanning information to predict and learn about changes in cell physiology over time in the bioreactor. So we showed in the last case study that changes in the ratios of different populations, in the case of the last case study, those were healthy and unhealthy cell populations, can cause a change in the observed dielectric spectrum. So now we're actually going to try and show how we can tie these changes back to the fundamental biology of what's going on. So this slide shows a common question that we get about dielectric spectroscopy, and that is, so that's great, what can these dielectrics actually tell me about what my cells are doing? How is this information useful to me in terms of biology? So really, in other words, it's easy to see how when some unknown electrical property changes that I can observe a change in my dielectric spectrum that I measure. So that's just a matter of physics as we covered in the beginning of the talk. But the harder and perhaps more interesting question is how are the biological changes which are occurring affecting the electrical properties of the cells, which I'm subsequently detecting by dielectric spectroscopy. So in this case study, we're going to show some data from a study that we've undertaken internally to try and answer that question for a specific change that we already sort of know is observable from the last case study, which is programmed cell death or apoptosis. So shown here are some light scattering data generated from the cells in case study 1. So as is commonly done, here a flow cytometer was used to classify the healthy and unhealthy cell populations in the bioreactor based on light scattering. So here you can see that the healthy cell population in the growth phase dominates, and then that population is roughly Gaussian, so that's shown as the green population there. But if we look at the same cells during the death phase of the culture, we can observe the healthy population is still present somewhat, but an unhealthy cell population has actually grown in, shown in red. And we can also see that those cells scatter light differently in general than the healthy cells. So these differences, as we saw before, correspond to shifts in the dielectric spectrum. So the question here is this apoptosis or necrosis? So we attempted to reproduce this shift to answer this question in the dielectric spectrum in a controlled apoptosis experiment. So in the experiment, 2 fed batch bioreactors were run in parallel, each with an identical fed batch process growing CHO cells. The cultures were monitored using an Aber-Futura frequency scanning dielectric probe. Each culture had its own probe, that is. So on day 14, the experimental culture, hang on a sec here, might need the experimental culture was treated with 1 micromolar staurosporine, which is a known initiator of apoptosis in mammalian cells. And so you can see here, this is the viable cell density growth curves versus time, that the viable cell density by the offline method drops precipitously after addition of staurosporine in the experimental culture compared to the control culture. The green box denotes this the window of the experiment that we're going to be looking at on the subsequent slide. And so shown here are the resulting dielectric spectra over time from those 2 cultures. So these curves are represented here as measured 1 per hour over the 60-hour time course of the entire experiment, and are colored rather through time from blue at time equals 0 to red at time equals 60 hours. And the curves are also normalized to each other from 0 to 1, as in the previous case study. So in the control culture, you can see that we observe very little shift in the dielectric spectrum over time, which is to be expected, as we only observe a slight drop in the viable cell density by the offline method at this point in the culture. In the culture where we added the staurosporine, however, which is shown on the right, we observe a rather drastic shift in the shape of the dielectric spectrum over time. And we observe this shift predominantly in the higher frequency end of the spectrum. So an interesting result, certainly. We're also able to confirm the formation of an apoptotic cell population, which increases in percentage of the total cell population over the course of the experiment by kind of pulling samples at representative time points throughout this time course. So you can see here, in this case, we did it by detection of activated caspase 3, which is a well-known marker of apoptosis in mammalian cells. So you can see that at the beginning of the culture, the healthy population, which is denoted by the blue peak, at the beginning of the culture is dominating when we only have a very small apoptotic population. As we move throughout the culture, we end up with a large apoptotic peak and a relatively small healthy peak. We've also been able to do some initial work, or additional rather, work by fitting population models to these dielectric spectra in order to allow us to quantify the percentage of the population which is in different cell states in the dielectric spectrum. And this is data that we presented actually at IFPAC a couple weeks ago, and I can forward that presentation on to anyone who's interested as well. So in conclusion, we showed how a rather drastic change in the dielectric spectrum can be observed over time when we induce apoptosis. And being able to detect a specific biological process like apoptosis has its own uses, such as perhaps being able to probe the causes of apoptosis in mammalian cell lines, or whether or not apoptosis inhibition strategies are even working. But looking forward toward the bigger picture, I think what this work really shows is the utility of dielectric spectroscopy as a tool to look at biological processes and their associated population dynamics in general. So other cases such as nutrient starvation, lactate overproduction, and other cellular metabolism changes have been linked in the literature to changes in observed dielectric spectra. So the work here on apoptosis not only lends some additional credibility to those findings, but it also demonstrates, I think, the promise of the technology in aiding us to probe these aspects of fundamental biology of the cells in real time in the bioreactor. So now I'm going to hand it back over to Lisa, who's going to talk about where we see this technology as really enabling some exciting improvements further downstream in the process and in the future.
All right, so thank you, Brandon. I appreciate taking us through the case studies. So I'm going to, as Brandon said, I'm going to transition the presentation now to look at how the technology fits into the overall process development paradigm. In other words, how does it couple with the other data that we generate during a process, and then how does that fit into guiding our process development and process control strategies? So to do this, I'm going to talk, as I said, through looking at the fundamentals around the bioreactor itself, and then how does it fit with process models and some key elements like signal processing and applied mathematics. All right, so in the industry, we all know there's a number of core competencies that we quite often have to bring to bear, and particularly biology and mathematics. And this slide provides a number of examples of other core competencies that are pretty critical in understanding what's really fundamentally happening, both from the cellular growth and productivity, but as well as the impact on the actual resulting product. So let's go one step further, and I want to put how these cogs fit together in context of the dielectric spectroscopy work that Brandon just showed. So if we start with the need to develop a technology that gives observability, and we want to end with what Brandon showed, an accurate and reliable online viable cell measurement, well, we need to actually go through a number of the different applied capabilities or competencies in order to actually get to the output that we want. And it's not just the dielectric spectroscopy alone, but it's also the elements of, let's say, metabolic flux models or CFD models or other important tools that we all tend to use when we're doing process development. So how do we go about building on what Brandon showed to optimize the experimental design? Well, we obviously have to run the right experiments, and we need to know what's happening with the biology. We need to manipulate the raw data to determine if the observable effects are in fact what we think are happening, and how does that tie back to the biology. And what was inherent underneath some of the slides that Brandon showed was some fairly significant effort around applied mathematics and signal processing. And then finally, how are those models used to interpret the physical meaning of the spectra? And of course, as we develop the models, we have to continually interrogate and refine them. All right, so let's take it one step further, and what I began this part of the presentation with was how do we really tie in the dielectric spectroscopy with the other technologies to get useful guidance of cell state and optimize experimental design. Well, what we've worked on here most recently is how to employ data automation and model development to take the dielectric spectroscopy data, couple it with the raw cell outline culture data. So in other words, what are we getting off of the bioanalyzer? And then any other offline context-dependent data. So what do we know about metabolomics or gene expression? And why do we care? Well, if we can be very creative about how we put all those things together, that gives us a very fundamental base then both real-time and near real-time for defining what the cell state is. So what are the mass balances? What are the key state of what those cells need to both grow as well as produce the product? From there, we need to reduce that to the right control scheme in order to tie it all back together. So of course, one of the things we have around the system is we have our dielectric spectroscopy, which is fantastic, sending signals and data to whatever repository. We also have the typical process instruments. We have potentially automated sampling data. We have the bioanalyzer. We have other offline tests. And as we're all running more and more bioreactors in parallel, we have more systems, more measurements, and more data sets that don't necessarily lend themselves to the analyses. So a lot of disparate time points and a lot of unique database structure and location. And one of the challenges we run into here is to get the most out of our dielectric data, we need to couple this more seamlessly with all the other data that we're getting. And so one of the tools that we've generated here is an in-house code that essentially compiles all that information and allows us in real time then to look at various runs, look at the various key culture data that we care about, and then calculate, for example, cell-specific rates or integral production trends. So the key thing here is the curves that Brandon showed are the result of not just computer programming to make such a user interface, but the actual science behind the interpretation of each of those curves and what it means. So if I click to this slide, another way to look at this is we take all that information that's giving us our observability that's so important. We're applying the ability to leverage all the computer programming expertise and applied mathematics to take all that information, pull it together, perform whatever signal processing needs to happen so that we spend less time manipulating data in spreadsheets and more time looking at what those key outputs are. What are these trends? They should look familiar. This is the graph on the right out of the case study 2 that Brandon showed. So how do we take and look at the data that's being generated more quickly, and where we can look at how the dielectric spectroscopy data coupled with bioanalyzer data gives us a better way of meeting the dynamic nutrient requirements of cell cultures, enables more responsive control, and finally allows us to take that knowledge and do a lot more with the process control. So as this section comes to a close, one area to consider around cell line selection is that as we are looking at the dielectric spectroscopy technology, either by itself or in conjunction with all the other modeling and offline bioanalyzer data, there's an opportunity to drive that capacitance-based technology further upstream and start looking at, let's say, clone selection or clone stability or some of those other key questions that our colleagues have earlier in the process. And so with that, we've reached the summary of our presentation. We showed a couple case studies. One, that how to make the measurements more accurate and useful in its application to predict the viable cell density or viable cell volume. In our second case study, we discussed the ideas around tying the real-time data to the actual biological changes. And as Brandon mentioned, the apoptosis-focused efforts is just a start in the research. There's quite a few areas that can go. And then finally, thinking about how dielectric spectroscopy fits in with the broader strategy for how to run a bioreactor and how to guide the process design. And so I really thank you for your time, and I will turn it over to our moderator for the Q&A session.
Thanks very much, Lisa, to you and Brandon. Yes, we have now moved into the Q&A part of our program. So at this point, I would like to invite you, our audience, to continue sending questions or comments by using the questions panel on the right side of your screen. So feel free to send in those questions. We do have a first question for you, Brandon and Lisa, and that is there any possible utility of the technique to aid in cell line development studies?
We think this is actually a really great opportunity.
apply DS actually in cell line development. So besides detection of cell growth and viability, which is a common selection criteria, obviously, we're also working on using the same technique, the frequency scanning part of it, to probe, I guess, population heterogeneity from clonally derived cell lines, which is an area of a lot of study right now, I think, in cell line development. So obviously, in order to implement that, we'd have to miniaturize the technology to the relevant selection volumes. Selection is not done at bioreactor scale. But one of the other things we want to look at too is how we can use this technology to probe how this population heterogeneity or this plasticity that we observe in CHO lines can affect the scalability of those cell lines as well. Using the technology to look at things that maybe aren't as obvious in the selection process. I don't know if Lisa, do you have anything to add?
No, I don't. I think you covered that well.
Okay, here's a question that just came in. Can you comment on the usefulness of DS for bacterial systems at high cell counts versus mammalian systems at more moderate cell numbers?
Yeah, so I'll start with the second half of that first. So for mammalian systems of more moderate cell numbers, it's all about signal to noise. So there is some minimum level below which we're basically not going to get enough signal to be able to detect or to be able to get at the useful data that we've shown here. That level tends to change a lot depending on the cell line. We've been able to get great frequency scans below 1 million cells per mil, for instance. But we've also looked at other cell lines where we've actually had a lot of noise up to about 2 million cells per mil. So all the data that we showed here was, I think, up about 4 million cells per mil. So we had a really good signal to noise in this case. So that's kind of just something to be determined experimentally. The second, or I guess the first part of the question was the applicability of this to high-density bacterial systems. And I think that's actually a great application of the technology, because you'll end up with actually a very high signal to noise in that case. One of the key watch-outs though there, I guess, is fouling of the probe. So the bacterial systems tend to have really high cell densities, can form biofilms, and so that's something to watch out for. But that's kind of true of any probe that you would stick in a bacterial bioreactor.
Okay, and speaking of bacterial, here's a question for you. Do you think this technology can be used with bacterial and yeast cultures?
So I'll take that one. So, I mean, the short answer is yes. And in fact, a lot of the early applications of the dielectric spectroscopy technology was in the industry around fermentation and brewing. So there's quite a bit of history there, and a lot of use of that across quite a broad scale. Is there anything more specific at all on that?
Yeah. Yeast and bacterial cell lines actually make for really good targets for DS because of the construction, the actual construction of those cells. They contain a cell wall, and they tend to have more drastic morphological changes, at least in the processes that we've seen that are associated with cell metabolism, and so that makes those metabolic changes easier to detect by dielectric spectroscopy.
Okay, here's another question. Is the discrepancy of DS and VCD observed in all mammalian cells?
No, actually. So we actually have one in-house right now where it behaves just fine unless the viability is below like 30%, which is most people would have harvested, I guess, below or before then. So no, we don't always see it, but it does happen, I would say, more than 50% of the time in our experience.
Okay, I think we have time for another question. What about non-spherical cells, such as those found in mycelial mats and fungi? How does DS deal with those?
So that's actually something that we've begun looking into. So I guess the question in that case seems to be whether or not the respiring and also the non-respiring part of the mycelium are actually contributing to the dielectric signal. So, you know, as we said before, the differences in the architecture and geometry of cell walls between these different parts of the mycelium might produce a sort of one of these two-term Cole models, as we showed in the deck. In theory, but this is an area actually that we're actively investigating right now.
Maybe to add on to it, I think it comes down to what is the fingerprint of the dielectric spectra with respect to the frequency scanning from low to high. And I think it would be some work around how to, you know, what's the baseline for fully functioning healthy microorganisms versus those in different states.
Yeah, sort of a range finding study needs to be done. Yeah.
Okay, I think we have time for one last question. Should one rely on SEDEX cell size numbers at low culture viability? Rustin Schenkman-Shire.
Yeah, so I guess we don't, but I wouldn't say that you shouldn't per se. So the way that we've been calculating our viable cell volume is by just taking the viable fraction of cells and then taking the average radius, I guess, of the cells as measured using a SEDEX, which is obviously not the most rigorous way of determining viable cell volume. We've shown that in the case of the case studies that we presented here that it seems to predict pretty well what the offline packed cell volume measure would actually predict. So we've shown that for that cell line it can work pretty well, but you know, I wouldn't say that it should or shouldn't rely on it, I guess.
Okay, Lisa, did you have anything to add to that?
No, again, I think Brandon did a good job.
Okay, well, thank you very much. That's all the time we have for today. I would like to thank everyone for attending today's webinar. Don't worry if you missed part of the presentation. Simply visit www.bendresearch.com where our webinars are archived. If you want to share this presentation with your colleagues, simply note their name and email address in the post-webinar survey and we will send them a link to the archived presentation. Please join me in thanking our speakers for today, Lisa Graham and Brandon Downey. You will notice that a post-event survey will pop up on your screen at the end of the webinar. Please fill out this survey because we would really appreciate your feedback as well as your suggestions for future webinar topics. Thank you all for your time. We hope you found